Afternoon. Good afternoon. Welcome back to CAIDPN 2020. Our next speaker is new to CAIDPN, but I have no doubt he will prove very popular with our members. Dr. Kevin Mitchell is an Associate Professor of Genetics and Neuroscience in Trinity College Dublin, and he's going to help us navigate the messy reality of genetics of neurodevelopmental disorders. Kevin, I'm struggling just to say that, but Kevin is the editor of a textbook on this subject and will help us understand the clinical implications of the recent advances in the field. This is a vital area for us in clinical practices. Advances in genetics and neuroscience inform our practice and refine the care we provide for children and young people with autism and intellectual disabilities. Having heard Kevin's TED Talk at TCD, Who's in Charge, You or Your Brain, I have no doubt he'll ignite your interest in the area. So again, we'll have a short time for questions after. Please post your questions, comments and reflections in the Q&A section. And there is a short delay in those coming through to me, so post them early in the talk. So thanks very much for joining us this afternoon, Kevin.
Thanks very much. Thanks, Heather. It's a pleasure to be here. I'm really looking forward to giving the talk and especially to getting questions and hopefully having some time for discussion at the end. So as Heather said, I'm going to talk about the messy reality of the genetics of neurodevelopmental disorders. And I guess I'll have to ask for the next slide, is that right? Thank you. So I won't have to tell this audience about neurodevelopmental disorders. Obviously, they affect a large percentage of the population. There's a very wide range in severity to the point where whether something is called a disorder or not is open for discussion, I think, in any particular case. They have a huge economic burden as well as obviously a personal one. And the reason is that there are lots of unmet clinical and educational and social support needs. Now, these conditions are largely genetic in origin, and that's been known since Hippocrates, at least. So we've known that they run in families for millennia. What we haven't known is what are the actual genes involved, but we're starting to learn that. And as we do, we're starting to reveal some of the biology which up till now has been very undefined. And of course, that's a reason why many of the clinical needs are unmet. So on the next slide, the goals of genetics are laid out here. Why would we want to figure out which genes are involved? So if we can identify the risk genes, that's the first step. The hope is that that reveals the underlying biology and that we can then translate that understanding to the clinic. And that can happen in multiple ways. Firstly, just by making genetic diagnoses instead of the very large sort of artificial constructs of diagnostic categories as they currently exist. The hope that that might lead to personalized medicine, where knowing that patient A who has a mutation in one particular gene might respond differently to a medication than patient B with a mutation in a different gene. Of course, that sort of approach has been extremely successful in cancer, in oncology, but not obviously applied much yet in psychiatric or developmental medicine. And ultimately, the hope might be that we could come up with new therapeutics based on an understanding of the biology for symptoms that require some sort of a therapeutic intervention, like new drugs. So the next slide, please. So that was the hope. And there's been tremendous progress, which I'm going to just give an overview of in genetics in the last 10 or 15 years. Absolutely amazing discoveries. We know so much more than we did 15 years ago. But the upshot is, depressingly, that everything is messy. The diagnostic categories that you all will know from clinical practice are highly overlapping in terms of symptoms, are also highly overlapping in terms of genetics. The distinctions that we've thought about between, say, rare and common disorders or simple and complex genetic inheritance turn out to be very fuzzy. Those dichotomies just don't stand up, actually. The relationship between genotypes and phenotypes is extraordinarily fuzzy. And the underlying biology is also not very clear. We get a general picture from the genetics, but maybe not as specific a picture as we had hoped for. And that may make us reconceptualize what these disorders are. that we're actually looking at. So on the next slide, please. The distinction between rare and common is where I want to start. So you'll all know that there are lots and lots of rare neurodevelopmental disorders like Fragile X syndrome, Rett syndrome, Down syndrome, and so on. Many of those have been known for decades or longer. They tend to have one specific genetic cause, which could be a mutation in a particular gene. It could be a deletion of a piece of a chromosome. It could be an extra chromosome like in Down syndrome, but it's one particular thing. And they have a spectrum of clinical manifestations, which can include things like intellectual disability and autism and epilepsy and so on. And then on the other side, we have the common category, which really means idiopathic. We don't know what the causes are, but we can treat them as a group. And so things like epilepsy and autism and schizophrenia have been treated and thought of in that way. So there's been quite a dichotomy between these two categories. And the ones on the left have a sort of a Mendelian inheritance, very simple in genetic terms. The ones on the right are thought of as having complex inheritance. And the next slide shows something about what that means. So for autism, for example, it's thought of as a complex disorder which doesn't have a single genetic cause, even though, for example, something like Fragile X syndrome can have autism as a consequence. Autism generally as a group does not all relate to mutations in that one gene. And complex disorders generally are associated with effects of multiple genes across the population, and sometimes in combination with each other, and sometimes in combination with lifestyle and environmental factors. And the next slide, please, looks at the inheritance of complex disorders, which differs from traditional sort of simple genetic conditions in that they cluster in families, but they tend to aggregate rather than clearly segregating. They often have high heritability, which means that genetic differences are the main reason or the main difference between who gets the condition and who doesn't. But it's also true that many cases are sporadic. So how can we square that circle? On the next slide, one of the ways to do that, to look at the genetic architecture of conditions like that, is to do things like twin studies. So if we look at identical twins who share all of their DNA versus pairs of fraternal twins who have 50% of their DNA variants in common, and we ask if one of the twins has a condition like autism, how frequently does the other twin also have that condition? And the next slide shows a graph for monozygotic or dizygotic twins showing these rates. So for autism, which is the sort of light blue bar there, if one twin has it, the other one has at least 70% and actually often more than that. Whereas if they're dizygotic twins, it's only about 10, it's under 20% anyway. And what that difference shows is that there's a really strong genetic component there. And also the very high rates in monozygotic twins show that those people who get these conditions are at very, very high genetic risk of having them. And we know that because we can look at somebody else who has the same genetics as that person and see this really high rate of the condition. So the next slide shows that you can work out what's called the heritability, which means how much of the variance in who gets the condition and who doesn't is due to genetic differences. And for autism, schizophrenia, epilepsy, they're all very high, higher actually than many things like heart disease or cancer or diabetes, for example. And also in these twin and family studies, you can see that there's almost no effect of the shared family environment, which really puts paid to ideas that were prevalent for a long time, that conditions like autism or schizophrenia were due to direct effects of parenting. There's no evidence of that whatsoever. The next slide shows that even beyond looking beyond twins, you can see the risk to someone who is related to a person who has the condition. So in this case, schizophrenia. If someone has schizophrenia and you are their sibling, you've got about a 9 or 10% chance of having it. If you're their cousin, you've got about a 2% chance of having it, and so on. So the risk extends across other relatives, not just in twins. And if you look on the next slide, what you see is that that risk also extends across disorders. So this is one of the areas where we're already getting messy. We've thought of these things as distinct from each other, and genetically speaking, they're not. So if you have a first-degree relative who has epilepsy, then you have an increased risk of having schizophrenia. And if you have a relative with autism or intellectual disability, your risk of schizophrenia also goes up. So your risk across all of the disorders goes up. There isn't clear water between them in terms of genetics. So the picture on the next slide, where we could think of these things as quite distinct from each other, is clearly wrong. And you guys know that clinically. The genetic picture, which is on the next slide, shows that these things are really highly overlapping with each other. Okay, so we know they're genetic, we know they're overlapping. Now what can we do about it? And the next slide starts to, oh, thanks very much. Yeah, you can add in epilepsy and intellectual disability. So disorders can be genetic, but in different ways. So you can have a condition which is a single gene disorder, like something like cystic fibrosis, where everyone in the population who has cystic fibrosis has a mutation in the same gene, and that circle around the Vitruvian man here is supposed to be the map of the genome, and the little red lightning bolt is where a mutation has happened. So everyone has the same mutation. Then on the next slide, you can have quite a different scenario where you can have a condition that everybody shares, like epilepsy or inherited deafness or retinitis pigmentosa, where different people in the population each have one mutation in one gene, but it's different genes in different people and different families. So that's called genetic heterogeneity. And the next slide just illustrates for retinitis pigmentosa, inherited blindness, how this can manifest and the types of pedigrees that you see. So this is different mutations in different genes in different families, but within each family, there's one gene to blame. And the next slide shows quite a different scenario where you go from a monogenic disorder to the, if you just press the button, please, and what's called an oligogenic one, where in a particular individual, having the condition is caused by not just one mutation, but mutations in multiple genes, to the next scenario, which is called polygenic, where actually it's mutations in hundreds or maybe even thousands of genes at once that push you over a sort of a burden of genetic mutations or variation into a risk threshold or disease state. And that's illustrated on the next slide, which just looks at a model. The idea is that we all have lots and lots of genetic variants that are common in the population. And if some of them increase risk of a condition, then there will be a normal distribution of that burden across the population. And some people shown at the right of this slide may be past a threshold beyond which that burden is too much and the system can't cope and they develop disease. I do want to emphasize this is a statistical abstraction, but it's a very useful model for thinking about polygenic conditions as well as quantitative traits more generally. So the next slide illustrates, it goes back to this dichotomy between rare and common disorders, and it illustrates two different ways of thinking about their relationship to each other. The first model, you can think that the monogenic cases are exceptions, while the ones on the right-hand side, the idiopathic cases, which far outnumber those other ones, represent what we could think of as real schizophrenia or real autism. And those will be polygenic. So they'll have a very, very different type of genetic inheritance. And that model has been the prevailing paradigm, I think, in psychiatric genetics for the past 15 years or so. But it's not the only model, and it's one that I and others have been arguing against for various reasons. So model 2 suggests that all the cases have some rare high-risk mutation. It's just that we don't know about them yet. And that real schizophrenia or real autism don't exist. They're not natural kinds. They're just artificial constructs that are diagnoses of exclusion. They represent our current ignorance, not a real biological category that differs from the other ones. And if you look in the next slide, where we're going to come to eventually is a unifying model, where most or maybe all cases involve at least one major mutation, but maybe several. But there are also real and strong modifying effects of the genetic background. So there's both genetic heterogeneity, different mutations in different people that you could say are primary cause, and there's polygenicity, that is the genetic background generally affects the manifestation of those mutations. So from a geneticist point of view and from a clinical point of view, this is extraordinarily irritating. This is like the worst case scenario for the genetics being tractable and for genetic discoveries having direct clinical applicability. But all hope is not lost. As we go on to the next slide, we can see that, first of all, there's been tremendous progress in finding rare mutations. So most of the conditions that I talked about at the start, like Fragile X syndrome, Down syndrome, Williams syndromes, and so on, were mapped and discovered. The genetic cause was discovered by simply looking down the microscope at chromosomes, where you could visibly see there's an extra chromosome or there's a piece that's broken off or something like that. Those are extremely rare to have such large effects that you can see. But there's a new method called comparative genomic hybridization, which gives you a molecular way to detect even very small deletions across the whole genome. So that's been very powerful to discover more and more of those types of conditions. And then the growth of and development of sequencing, so genome sequencing, has been phenomenal over the last 20 years. And that gives us a chance to look actually at all the mutations that we can see, at all the genetic variation we see across the genome, and try to find ones that are associated with disease. So the next slide shows, just to start with these big mutations, these genomic rearrangements, things like deletions or duplications, inversions, insertions, translocations. There are lots of things that can happen when chromosomes are pulled apart. Sometimes that doesn't happen right. And there are certain sites in the genome where, because they have repetitive sequences, the machinery gets a bit confused, and you can have deletions or duplications, for example, that happen at that site with a recurring frequency across the population. And what that means is that you can find many instances of people with effectively the same mutation, like a 22q11 deletion, for example. When you do that, you can assess statistically the association of that deletion with any sort of medical condition. Many of those arise de novo. And what that means is they're not inherited from a parent who has the deletion or duplication. They arise in the generation of sperm or eggs. And that's a really, really important factor here. The next slide shows just this molecular method of genomic hybridization that lets you find these things. And the slide after that just illustrates one of the first places where this was applied, which was actually in autism. It was developed as a tool to look at cancer in the first instance, but Jonathan Sabat and colleagues tested in autism if these kinds of rearrangements might carry a risk. And what they found was that indeed people with autism have a higher rate of de novo, what are called copy number variants or structural variants, than people without. And on the next slide, what they found, which also makes sense, is that that higher rate of de novo mutation was only seen in sporadic cases, not in inherited ones, which is what you would expect. If it's in a family where autism just pops up in one individual, that's much more likely to be caused by a new mutation than a family where multiple people have it. That's more likely to be caused by inherited mutation. But actually, even in the inherited ones, you can see that sometimes those are caused by inheritance of a structural variant like this. And the next slide showed a real surprise in the field, which is that when people started to look for these associations across different conditions, they kept zeroing in on the same ones. So the 22q11 deletion, for example, was found in people with schizophrenia, with epilepsy, with autism, with intellectual disability, and so on. And there's a whole growing list of these kinds of genomic rearrangements that, if you look at the pie charts at the bottom here, occur in people with different kinds of conditions. And generally speaking, each of them can manifest in many different ways. Now, sometimes, for example, the 15q13.3 deletion has a much higher rate of epilepsy than other deletions do. But you'll still notice that the expression of each of those is a highly variable spectrum of clinical symptoms and conditions. Next slide, please. So those kinds of structural variants contribute to many neurodevelopmental disorders. You can get evidence for them because they recur. They're often de novo, but sometimes inherited. They have more severe effects in males, which is something we can come back to. And interestingly, they account for a greater percentage of cases of more severe conditions. or presentations. So in autism, for example, a greater percentage of cases of autism can be attributed to those kinds of genomic rearrangements than in bipolar disorder. And the next slide shows a nice illustration of that. This looks a little bit complicated, but up at the top, you can see this gradient of neurodevelopmental pathology with mental retardation or intellectual disability and autism and schizophrenia on the left-hand side. They have a greater pathology versus things like bipolar mood disorder on the right-hand side. And down at the very bottom, what you'll see is that the DNA structural variants I've just been talking about are much more common in those ones on the left-hand side of the slide. And there's an evolutionary reason for that, which is that when these conditions arise, when these mutations arise and they cause a serious condition, then it's very unlikely for those people with that condition to have offspring. And so in effect, the variant is very rapidly selected against, as is the case for Down syndrome, for example. So, so the severity of the condition is sort of associated with how likely it is to be caused by one big mutation as opposed to multiple mutations. Next slide, please. Okay, so what about point mutations? Just a single change in the DNA sequence. Well, these arise every time we make sperm or eggs. Some mutations creep in. There's at least 70 mutations every time we make one. So over generations and generations, of course, you will accumulate more and more genetic variation. And we'll have some new ones. Each of us will carry some new mutations, which will be rare in the population. And then we'll have this vast array of older ones, which are common. And now you might ask, well, how do we ever survive if this genetic variation is just accumulating? Well, it's counterbalanced by natural selection, who's trying to remove damaging variation. So the next slide shows a kind of a balance between mutation and selection. In one generation, mutation creates variation, but unfavorable mutations are selected against because those individuals don't pass them on because they don't have offspring in the most severe case. And then you have reproduction, and then you have more mutations come in, and this just goes on and on and on. And if you just click the button, please, what you see is that new mutations enter the population each generation. Some of them cause disease that they're rapidly selected against. Some of them just increase the probability of disease a bit, and they're selected against, but more slowly, so they can persist for a while. And then if others that have very tiny effects or no effect can just persist in the population forever, basically. So the next slide shows a picture of genetic variation where, if you look at the bar on the bottom, you have new mutations that have just arisen in the sperm or egg that generated you. Your parents don't even carry them, but then you've got some other ones that are in your sort of family clan, your family tree, and then further back you've got ancient ones that arose a long time ago that are common in the population, but that have very small phenotypic effects. And the next slide just shows in any individual, about the number of these. So we might each carry about 70 de novo mutations, about 300,000 rare inherited ones, and about 3 million common genetic variants. So all of those can contribute to disease. And the next slide just illustrates that in order to find those, we need to sequence the genome. Thankfully, the cost for sequencing the genome has plummeted from the original, which cost $3 billion to sequence one genome, to about $1,000 to sequence a genome now. And the next slide just shows one of the ways that you can do that. You can sequence the whole genome now. People started by sequencing just the coding regions of the genome, just the 3% of the genome that actually encodes proteins. And when you do that, then you can start to look, like on the next slide, for mutations in particular genes that are arising in people with these different kinds of conditions. And when people did that, they found lots and lots of these genes. And as with the copy number variants, one of the striking things is that most of the time they overlap. They're found in people with schizophrenia and with autism and with epilepsy and with intellectual disability. So the boundaries between those conditions from a genetic point of view simply blur or disappear altogether. Now the next slide shows just a couple other examples. This is a nice paper that came out recently in autism, and their results are shown on the next slide. There's a bunch of genes that what the bottom of this is just a plot by chromosome, so where in the genome the gene falls. and the vertical axis is the statistical evidence for association. And of course, that statistical evidence is relates to the sample size. So in order to get the evidence for these rare mutations, you have to have tens of thousands or even hundreds of thousands of people sequenced, which is in the area where we're getting to now. So there's loads and loads of genes implicated in autism. The next slide shows something similar for schizophrenia. And in fact, many of these genes overlap between these different conditions and with developmental disorders generally. And the next slide graphs the frequency in the general population on the right, on the x-axis, with the odds ratio or the increased risk of having the condition on the y-axis. And what you'll see on the very left there at the top is that the ones that have the highest risk of the disorder are the rarest in the population, which makes sense because they're constantly being selected against. The only reason we see them is because they keep recurring over and over again. And then on the right, those blue dots over there are all the common variants that have been associated with schizophrenia, which have tiny, tiny effects individually, but can have an important effect collectively. So the next slide just shows a kind of a re-evaluation of this distinction between rare and common disorders. So the idiopathic cases, back in 2000, there was a great big pool of those, but cytogenetics had already shown us a bunch of things like Fragile X mutation and Rett syndrome and so on, where we knew of those rare syndromes. But when we could do the comparative genomic hybridization, we could find more and more. And I'm using the royal we here, by the way, for the whole field. And as we can do exome sequencing and now whole genome sequencing, we're finding more and more single gene mutations, which means that the pool of idiopathic cases is shrinking. And in fact, it may shrink to zero if we find out all the single gene mutations that are associated with that. The next slide shows if you click the next button, please, that a way of reconceptualizing this is that those common idiopathic categories are simply umbrella terms. We just didn't know that they were composed of all these other single gene mutations, or we didn't know what they were. And the next slide just illustrates that if we think of, because these disorders overlap so much, we shouldn't think of a mutation causing autism or schizophrenia. We should think of it as causing something that's been referred to as developmental brain dysfunction, which can manifest in different ways. So each of those mutations is very, very rare, and they're strongly and rapidly selected against, but the conditions are collectively common. And what that means is that there must be a very large mutational target. That is, there must be many, many genes that you can mutate that will lead to this kind of manifestation. And that's a real mystery, why that should be. Why should there be so many genes that can be mutated that would cause you to have psychosis, for example? And we'll come back to that later on. Next slide, please. Ask a different question. If there are these single gene mutations that have such high risk, why don't their inheritance patterns look more Mendelian? They're so variable, their effects are so variable. So something else has to explain differences between patients who carry the same mutation. And there's other sources of variance. They include other things in the genetic background, potentially environmental variants that will differ between people, but also intrinsic developmental variation. And we'll look at genetic background first. So the next slide just shows that there's nothing strange about this. We shouldn't be surprised by genetic background effects. They're completely ubiquitous, even for single gene disorders like cystic fibrosis. So in the middle here, we have the CFTR gene. If you have a mutation in that, a pathogenic one, you will get cystic fibrosis. And if you have cystic fibrosis, you will have a mutation in that gene, more than likely. But there are also mutations in other genes that affect how severe the phenotype is and which organs are affected more than others, for example. And so that's a very typical thing, and the same thing will affect these mutations we've just been talking about for psychiatric phenotypes. Excuse me. The next slide, please, just shows a model of this. So the answer, if we're talking about the genetics of these conditions, is that there is not one answer. There's lots of different mechanisms and architectures at play all at the same time, and we just need to tease them out. So you can have some cases that present like the scenario on the left, where there's one mutation, it's really high penetrance, so it causes the phenotype, some specific phenotypic profile, very, very robustly. And those are often sporadic. The reason they're sporadic is because they get selected against very quickly. Then you'll have other situations like on the far left where you can have 2 mutations, but they could both be rare, but together they cause the phenotype. And then you'll have scenarios like in the middle where you could have 1 big mutation or maybe a couple, but there's also a sort of a big broad background of genetic modifiers, a polygenic background, and then you can get very variable phenotypes. So all these things are happening at the same time. And the next slide shows an interesting observation. So if you look at, this is what's shown on the left is a list of different kinds of deletions across the genome. Not different kinds, different specific deletions that have been found. And when you look in cases with those, what you can see is that the ones that are most severe by themselves usually arise de novo. That's in one of the middle columns there. And if you look to see, do they have a second hit somewhere else in the genome? Do they carry another structural variant? Then those patients normally don't. The reason is they don't have to. In order to be a patient, that 1 mutation is sufficient to cause that. Whereas down at the bottom, you'll have some like 16p 12.1, where it's very rarely de novo. It's much more common for it to be inherited. And in those cases, you'll see that the percentage of people who have a second hit, another big mutation somewhere else in the genome, is much higher. And that's simply an ascertainment bias in a sense, because in order for them to be a case, they have to have another mutation somewhere. The 16p 12.1 deletion isn't sufficient by itself. And the next slide will look at not just multiple rare mutations, but this other question of the modifying effects of the polygenic background. So common genetic variants can also influence risk. Their effects are tiny individually, almost negligible, but their collective effects may be considerable because there's so many of them. So how can we find those? Well, the next slide just shows again how many of these are in the genome. So 3 million each of us carry, where at a particular site, as shown on the next slide, there may be these colored ones in the middle here. There may be an A or there may be a T. So whereas the rest of the genome is typically the same between most people, those variable sites, could be 30% of people have an A, 70% of people have a T. So we can scan across the whole genome and we can ask, are there sites like this where one of the variants is more frequent in people with autism or schizophrenia or intellectual disability, whatever it is, than the other one? And that's a pure epidemiology. It's like asking whether smoking is more common in people with lung cancer, except that in this case, the risk is much tinier than that. So we have to look at sample sizes of tens of thousands or again, hundreds of thousands. And people have done that now very successfully. As shown on the next slide, you can use these microarrays to do this. And the next slide, please, just shows the sort of pathway that you use or protocol to compare cases and controls and look for these tiny differences in frequency, which illustrates some effect on risk. And the next slide showed a paper that was really an amazing step forward, looking for these kinds of effects for schizophrenia. And they found at the time, this was 2014, 108 associated loci. There's many, many more than that now. But the next slide shows this, a similar plot to the one we saw earlier. So chromosomal position on the bottom and the statistical evidence for association on the x-axis, sorry, the y-axis. And so each of those points in this Manhattan plot, each of the skyscrapers, is a site in the genome where there's some associated common variants, but their effects are tiny. And if you just click the button, what we can see though is that you can add up for any individual, like we'll all carry some of these common variants, but some of us will have more than others. And you can add up how many risk variants we each have, and that's called a polygenic risk score. And then you can ask collectively how much of the variance does that explain? And in this case, it explained about 7% of the variance across the whole population. So the next slide, please. Just summarizes a ton of work by a ton of people in the field looking at this. And the main points are this. First of all, the polygenic risk, just like the rare mutations, is not specific to any given disorder. It's very much shared across disorders. It even correlates negatively with things like intelligence and educational attainment. So there's something sort of broadly going on there. It's not just a disease-specific effect. There's thousands of genes implicated, like the ones that are rare. They're enriched for nervous system expression. That's a good reality check. We'd be worried if they were pancreatic genes or liver genes. So they're brain genes. Many of them are expressed particularly in the fetal brain, which is, again, a good sign for neurodevelopmental disorders. They have very, very diverse functions. So they don't really converge on specific biochemistry, but they're all broadly enriched. Sorry, they're not all. They're enriched for neurodevelopmental genes. So one way to think about this is that there's nothing specific about this for particular disorders. It's a very generic, shared kind of risk, and that it may reflect the general neurodevelopmental robustness. And the idea is that rather than causing disease itself, it may modify how well you can buffer, or a person can buffer the effects of a rare mutation. So that may explain why some people with a rare mutation would get a disorder while others would not. And the next slide just shows a sort of a schematic that moves on from just thinking about these rare mutations and now sort of filters their effects through this polygenic burden. And again, there's a sex effect there that we could talk about. So what you end up with is an overall genomic risk, which goes back to the picture that we initially saw of the monozygotic twins who have very, very high genomic risk for these conditions if their co-twin also has them. The next slide, please, just shows one nice paper which fits this idea of the two types of risk, rather than occurring in different sets of patients, co-occurring in the same patients and interacting. And what this shows is that if you look just on the top there, that what's on the x-axis is the odds ratio, or the risk of a particular rare variant that a patient is found to have. And what's on the y-axis is their polygenic risk score. And what you can see is that the ones with really high odds ratios over on the right do not have particularly high risk scores. And the reason is they don't have to. Those mutations are sufficient by themselves to cause the condition. Whereas the ones towards the left, which they only increase the risk, 5-fold or 3-fold, they also have a high polygenic risk score. And together, those give the overall genomic risk. Next slide, please. Okay, genomic risk is not the whole story. These disorders are highly, but they're not completely heritable. And what's inherited is actually not a disorder, it's risk for the disorder. So there's some non-genetic sources of variance. The next slide asks the obvious question, maybe this is environmental. Well, as I said, that in twin studies and family studies and adoption studies, there's very little evidence for any environmental effect of the family environment. And people have looked for other environmental factors as well, and really there's very little there that's in any way kind of systematic that can explain the rest of the variants that we're looking for. So there's something else that we're missing. It's not genes, it's not environment, it's something else. And the next slide illustrates what that may be. The risk is inherited, not the disorder. So how the risk plays out depends on how development plays out. These are developmental disorders. So the way that the brain develops is ultimately what we're interested in. And the genetics can give a risk for developing in one way or another, but whether it actually does or not in an individual depends on some randomness. Development is a noisy process in engineering terms. And the next slide just illustrates the journey that you have to go on to explain how you get from genes to psychological traits. You have to go through development. And if you just click on, please, and to the next slide, brain development is just extraordinarily complex, and it's also very noisy. So the genome encodes biochemical rules of how the brain should self-assemble, but it doesn't encode specific outcomes. The whole process just has to play out. And you don't get the identical result every time you run that program. And we can see that in the physical form of our bodies. So the next slide shows identical twins who at first glance really look very similar. But if you look a little closer, you can see, well, they're not exactly the same. You can tell them apart. There's some variability there that doesn't come necessarily from experience, it's just from the way that the program played out. And actually, you can see that in each of us. The genetic program plays out twice in each of us on either side of our body over embryonic development. They develop largely independently of each other. So the next slide topical, shows one way to see this. If you take a full-on frontal selfie of yourself and split it into left and right sides and make mirror images of those, if you click on, please, you can see that they look quite different from each other. So that shows that this variation is just there. It's not coming from outside, it's from inside. And if you click on, please, that will act to sort of spread out the range of phenotypes that are associated with any given genotype. That's true for everything. It's particularly true for these disorders. And the next slide shows, well, it illustrates a point, which is that in brain development, that doesn't just contribute to a spread of quantitative traits. It can contribute to qualitatively distinct outcomes by virtue of the fact that that brain development is a highly nonlinear process. Every step is contingent on prior steps having happened properly. So one small change at one point can cascade over the rest of development. And if you click on, please, this schematizes the idea. So you've got an organism that's developing along a certain trajectory, and the landscape which it's rolling over is meant to illustrate the particular genome that we each have. In this case, those two genomes are the same, the two genotypes, but just due to very small effects at one point or another, development can be channeled down one trajectory or another. And if you just click through the next few slides, I don't have time for them, you can see that in brain anatomy. If you click on, please, and one more, and again, just to give you the overall picture, that overall genomic risk that we talked about is again filtered through developmental variation to give some specific outcome in each individual patient that we're calling developmental brain dysfunction that can manifest in these very different ways. Okay, next slide, please. Now, we did all this genetics because we wanted it to tell us about the biology. What has it told us? Well, there's enrichment for genes involved in neural development. We sort of knew that. These are neurodevelopmental disorders. There is disappointingly no convergence on specific biochemical or neural pathways. And if you go to the next slide, please. That tells us something important. These conditions have genetic origins, but they don't have proximal molecular mechanisms. They're not like cancer, where the mechanism is really genetic dysregulation. They have neural mechanisms that emerge over development. So we have some interesting, important questions to ask. Why do mutations in so many different genes lead to those conditions? Well, it's not because there's lots of genes that control hallucination or perception. It's not because there's lots of genes that control sociability. There's lots of genes that are involved in building a brain. And when there are disturbances in neural development, there are ways that the brain gets channeled into these certain states that manifest as the conditions that we see. So the functions of the genes here don't relate to the symptoms that emerge at all. We're not going to learn anything at this stage by looking at what those proteins do about the ultimate symptoms that we see in the people with these conditions. And that's a stark kind of reality check, but it means that the genetics is just the first step. We have to go through all of the neuroscience to figure out what's coming next. Can you go to the next slide, please? I'm going to skip on a few slides here, if you don't mind, and one more, and another, and again. This is a useful concept. The idea that in a developing system, especially a dynamical system, you can have certain states that act as attractors. That is, the brain may be able to be in certain states, and the sort of normal range of functioning is one of those states that most of us thankfully end up in. But you can also have other states which are new. They're not designed by evolution. They're just in engineering terms, and my apologies if this sounds like an offensive term, it's just an engineering term, are called a failure mode of a dynamical system. So there's something novel that emerges from the way the system is put together so complicatedly, and those may manifest as these qualitatively novel states, like psychosis or mania or depression or anxiety. Next slide, please. And one more. Okay, so that's what I just said. It makes us reconceptualize the relationship between the genotypes and the phenotypes. There is no direct relationship at the molecular level. We have to tease it out through all of the neuroscience. And next slide, please. This just illustrates the really sort of long, complicated trajectory from a mutation to emergent psychological symptoms and what all of the steps we would have to fill in to understand, to have an explanation for why that mutation made a person think that the CIA was tapping their phone, for example. Okay, next slide. Just to recap, everything's messy. The genetics has been amazing. We've identified hundreds of genes, and that's fantastic, but it didn't provide the direct kind of clean answers we might have hoped for. Instead, it paints this really messy picture about these different kinds of categories, the relationship between genotypes and phenotypes, and the underlying biology being quite indirectly related to the symptoms. So what does that mean for clinical implications? The next slide, that's what you guys are here for. I listed at the start the possibilities here: genetic diagnosis, personalized medicine, new therapeutics. And there have been some advances, but really not much, frankly, from the genetics, not yet. It is possible to make a genetic diagnosis for growing numbers of patients, and that can be useful in terms of giving some certainty. It gives surety to patients and their families. We're beginning to be able to look at rare conditions, at many, many patients with them, to determine whether, are there some prognoses we can make? Do they tend to have this condition or that? Maybe they would tend to respond to one medication or another. There's relatively few success stories in that area yet, but it's very early days. As far as new therapeutics, there's really nothing that comes out of the genetics that directly suggests a therapeutic action in the way that we've seen in cancer. And that's not a surprise because the phenotypes are so far removed from the actions of the gene. So it's going to take a massive, massive amount of work to get to new therapeutics out of the genetics. And the next slide, that's just a review that we wrote on how you could do that in animal modeling. And the next slide gives an idea of how you might, sorry, you can just click through this whole thing here just to bring the whole thing up, please. And just one more, please, and just stop there for a sec. What we'll have to do is, look at our patient cohorts, identify the disease-causing mutations, and if you look to the left, that may let us stratify patients. If you look to the right, we can make mice with those mutations, and then we can start to do some neuroscience in them and see, well, what happens in the brain when you have that kind of a mutation, and which systems are affected. And ultimately, if you click one more, at that level, you might be able to figure out, aha, I see this system is affected. I can tweak that with a drug that affects this ion channel that's expressed in these neurons in this circuit that mediate anxiety, for example, or something like that. But we're years away from that. The genetics doesn't tell us that directly because these are not proximally molecular conditions, they're neural conditions. So there's a long way to go. I'm hopeful about it, but we'll have to be realistic about what the genetics can offer. And the next slide, please. That's just a plug. So the book on the left is an edited academic book on the genetics of neurodevelopmental disorders. The one on the right, which came out in 2018, is intended for a broader audience, but does look at the origins of our psychological traits in general, but also these kinds of neurodevelopmental disorders. So I will leave it there, and hopefully we have time for a few questions at least. Thank you very much for your attention.
Thank you very much, Dr. Mitchell. A lot of questions and comments, so we'll go through some of those if that's okay. It's great to have that sort of insight into everything that lies behind the reports that we're getting from our clinical genetics colleagues, and someone commented about how sobering it is to know how much we still have to learn. The first question is from Dr. Tom Burney. Given the tendency for research studies to seek diagnostic categories, such as ASD and ADHD, rather than symptoms, does genetics support the validity of these disorders as entities, or are they an artificial result of our perception and research? Then secondly, 8 years ago, a study suggested that ASD might fragment into two components, dominated by impaired sociability and RBS symptomatology. Where has this got to?
Yeah, so I would say that genetics absolutely undermines the validity of those categories from an etiological point of view, at least. It's clear that the risk for them overlaps genetically. Now, That doesn't mean that the endpoints couldn't have some validity from in terms of underlying pathology as a state in which the brain can end up that could be more or less distinct from another state in which the brain could end up. So the genetics doesn't necessarily trump the clinical findings or categories, but it gives a different lens to view them. And certainly given how much debate there is about clinical overlap between them, I tend to see the categories as artificial constructs. They're clearly not natural kinds, at least. They have, they're clusters. In some cases, they're forced more than others. The distinction between schizophrenia and bipolar disorder, is a debate that goes back 100 years, and it's ongoing every time there's a new DSM edition. So I think the genetics, shows that variability and overlap. The second question about subtypes, I think the genetics has not shown those subtypes. We've seen any individual mutation, and even monozygotic twins can have quite different presentations clinically. So I don't think we can look to the genetics in the hopes of it really giving different subtypes of these conditions. There's just not a close enough relationship to the emergent symptoms from the different molecular causes in the first place.
Thank you. So it's really challenging some of our previous thinking. Dr. Mark Lovell wants to ask, do you think that we might get to a point where we can identify ASD that is genetically caused? And if no genetic cause is found, can reframe it as a post-traumatic or environmentally caused disorder, attachment or developmental trauma, if there is evidence of sufficient adverse childhood experiences? We often have a debate between the two.
Yep. Well, so first of all, the evidence for a genetic component or contribution to the etiology of autism is overwhelming in my view. It's one of the highest heritabilities of any common condition. So 80 to 90% probably of the variance in who gets it is directly attributable to genetics. I think a lot of the rest, given there's no evidence for family environment effects, is probably caused by developmental variation. So we're already at the point where we can identify in many patients with autism a major contributing mutation. And I don't know what the total yield is these days, from the clinical genetics, but it's up in the 30% kind of range. Whereas, for intellectual disability, it's probably 70% these days. So the reason it may not be higher than that is that many of the cases are complex, genetically speaking. They involve multiple mutations, then it's harder to find them. And so that whole picture, there isn't really one answer, but we already can find lots of mutations there. You wouldn't diagnose someone as having autism on the basis of having the mutation. The diagnosis of autism is still a clinical diagnosis. The genetic diagnosis doesn't override that or supersede it. It's a complementary diagnosis about the etiological origins within each patient. And I see those two things as being, they'll both still be useful going forward. Sorry, Heather, you're muted there.
Sorry. Yeah, no, sorry. I just said I think that's a really helpful way for us to think about it clinically. We have an interesting question from Dr. Ashley Liu, who's a neuropsychiatrist. Why do I still see an abundance of published literature investing genetics of endophenotypes like cognitive models? Also, what is the future for pharmacogenetics in neurodevelopmental disorders?
Oh, those are great questions. So the endophenotype model is based on the idea that while the genetics of the conditions may be really complex, maybe you can break them down into their subcomponents of symptomatic dimensions. And maybe those dimensions will be closer to the action of the genes and their genetics will be simpler. So for example, in schizophrenia, maybe you've got some genes, some genetic variants causing a defect in working memory. You could have other genetic variants causing a defect in sociability or mood or perception and so on. It's a reasonable hypothesis. It turns out not to be supported at all by the genetics. The genetics just doesn't break down like that. The endophenotypes aren't any simpler. They don't segregate that way. The genetic variants don't align with one symptom or another. So that picture has been tested really robustly, I think, and in my view, shown to be false. That doesn't mean everyone who's been working in that, taking that approach is willing to give up on it, because, well, you can do those experiments, you can publish papers on it. The pharmacogenetics is interesting. I think that pharmacogenetics has been one where there are lots of big effects, and that's partly because our responses to drugs, to things that we've never been exposed to in human history, can be very large, even from common genetic variants, because they haven't been selected against for that reason. It's a new effect. So pharmacogenetics very generally is successful in who metabolizes which drugs. It hasn't been successful in psychiatry so much in who responds to one drug or another. There's some few examples in epilepsy, maybe in Dravet syndrome caused by mutations in a particular sodium channel. The typical anticonvulsants are contraindicated in those patients. So the genetics there directly informs the drug treatment. That's a really rare scenario. Hopefully more and more of those scenarios will come up as we accumulate more data. You need to know the genetics in the first place to know if there's a difference, to see a difference in drug responsiveness.
Thank you very much. And yes, certainly Professor Beefsag has commented, for people can see there, about the drug treatment of epilepsy. We'll give the final question to Claire McKenna again, given that she recommended that we invite you along to speak, and just highlight that people can read an interview where Claire interviewed Kevin in the BJ Psych Bulletin. So a bit more of what he's just been talking about, actually. But Claire wants to ask, how much hope is there for a polygenetic risk scores predicting prognosis for individuals with neurodevelopmental disorders?
Yeah, that's a really interesting question. So there's lots of people who are hoping that polygenic risk scores will be clinically actionable. I find that unlikely for two reasons. One, they don't capture that much of the variance. Well, let's say multiple reasons. Two, the way that they act is not, you can't really separate it from the rare mutations that we each carry. So, I might have a high polygenic burden, but it can have no effect in me if I don't have a rare mutation that would cause the condition. And so by itself, it's not really very predictive, either statistically or biologically, although there's lots of people wanting to use it. But the other thing is that it's not clear what action you would take. If you find that from polygenic risk, someone's risk of schizophrenia or even overall genomic risk is higher than somebody else, well, what will you do with that information? It's not like you're going to prophylactically prescribe antipsychotics to people who aren't psychotic, right? And then so if it was heart disease, great, you can take all kinds of lifestyle and medication factors that could ameliorate a known risk without themselves being a risk. That's not, as far as I know, the case for psychiatric conditions. So it's not clear what usefulness those things would have, even if they did have some validity as predictive factors, which unfortunately I think they don't. And can I just say one final thing? I actually think that the closest application of genetics in this field in the clinic will be it'll be in the fertility clinic. It'll be in reproductive choices, that's where a prediction is in a sense actionable, and that carries all kinds of ethical concerns. But it's already happens, of course, for many, for Down syndrome, for example. There's no real sort of sharp line between Down syndrome and many of these other conditions that we've been talking about. So they're likely to inform reproductive choices, and that probably is the closest application in the clinic that we'll see from this type of genetic research.
Thanks very much, Kevin. I think my mind's a bit blown, but really useful for thinking about what we should be thinking about now in our clinical practice and what the future might hold. For us, you've very kindly agreed to stay on and answer some more questions in the Q&A, so that's great.
Yeah.
We have a break now for just about 15 minutes. And I'm really excited that Professor Gielberg is joining us this afternoon. Since I first read about his ideas about essence, I've been waiting for about 10 years to hear him speak in person. So I can't believe it's just another 10 minutes, and that'll be our final talk of the day before some closing remarks. So we've got a break now, and see you again at about a 2:45. Thank you.